Welcome, everybody. My name is Jeff Gray. I'm a professor of chemical and biomolecular engineering at Johns Hopkins, and I am the director of the Rosetta Commons and the director of this research in mentoring for Post-Baccalaureates program that we have in biomolecular structure prediction and design. In this webinar, we will be talking about the program, telling you a little bit about the science that's involved and the opportunity to basically do a year of research in a leading lab as part of the Rosetta Commons and give you a chance to ask questions. If you want to type questions into the Q&A, I invite you to do that at any time. We may save some to the end or we may do some as we go. I also invite you to go ahead and chat as you go. If you feel comfortable, I'd love for folks to introduce themselves, where they are coming from. And if you want to share anything interesting about yourself, please go ahead. We'll let the chat go to keep it interesting. So at this time, I want to go around the room and let my team introduce themselves. Let's start with Matt.
Hi, everyone. I'm Matt O'Mara. I'm an assistant professor at the University of Michigan in the bioinformatics department. And I'm a co-director of the RAMP program, and I've been involved with the Rosetta Commons since I was a postbac before my graduate school. So I'm absolutely thrilled to be here and participating in this project. And I'm looking forward to another good year of postbac program.
Camille?
Hi, everyone. I'm Camille Mathis. I'm the Director of Operations for Rosetta Commons and the Program Manager for the RAMP and the REU programs. Thank you for joining us today.
And Stephen.
Good afternoon, everyone. I'm Stephen Thomas. I'm based at Michigan State University, and I'm here to work with you all in terms of the GFP grant writing workshop, as well as the mentor training. I've been making sure that it's cohesive interaction between you and your mentors throughout your time in grant.
Great. So I will dump right in and get us started. First, you might be asking, who is the Rosetta Commons? You're looking at a picture of a lot of people in the Rosetta Commons. The Commons started a long time ago now, maybe before you were born. A lot of us who initiated it were studied in David Baker's lab. You might know David Baker's name as he just won the Nobel Prize in chemistry this year. And a lot of us trained in his lab. And as we went out and started our own labs, we formed a collaboration, which we called the Rosetta Commons, to work on the computer code together that does protein structure prediction and design. And so over the years, this community's grown as the scientific advancements have just built upon each other and allowed so many things to happen. So now we're this great big group. There's hundreds of developers, these different labs that are part of it. We have annual conferences multiple times a year on different continents around the world. And there's lots of shared code in the Rosetta repository, and that is the beginning of everything. The next slide shows us how these labs are distributed, mostly in North America and Europe, and a number of labs now in China. The RAMP program is also distributed over these labs. If you go to the next slide, we have 9 or 10 sites that have volunteered to host a postdoctoral scholar next year. And so when you apply, you'll get to state your preferences about which scientific areas are most interesting to you and what locations you might want to go to. All right, so let me tell you a little bit about the science to get you excited. So the Rosetta community has been working on protein folding for a long time, since the late 1990s. I was in David Baker's lab from 2000 to 2002. And what I'm showing here is protein folding, and the genome has all of the genes, and each gene is a sequence of DNA nucleotides that you can translate into protein. And then those protein sequences fold up into three-dimensional shapes. A grand challenge for biology that's been recognized since Linus Pauling's time is, could we take the sequence of a protein and figure out what the structure is that it folds into? And you might want to know that because the structure determines what it does. Is it going to bind to something? Is it going to hold something together? Is it going to make a cellular structure? Is it going to be involved in control or cancer or signaling or transport or membrane regulation? So really the structure of a protein is the best way to view it and understand its function in biology. And since the late 90s, there would be these competitions, these challenges called CAST, the Critical Assessment of Structure Prediction. And what those lines are showing is progress every 2 years. As the line goes up, we're getting better and better at predicting. And a lot of that progress is the Rosetta code, as the Rosetta code made a lot of the first breakthroughs and got the first structures right. And then the last few, CAST 13, 14, and we had CAST 15 last year. CAST 13 is AlphaFold 1, which was a machine learning, artificial intelligence approach. And then the green line is everybody in CASP 14 except AlphaFold 2. They all did better than AlphaFold 1. And then CASP 14, AlphaFold 2 got almost everything correct. On the right, you're looking at the two colors are structures of the experimental structure of the protein and then the predicted structure of the protein. You can see there's so many things that are just right on, and that's super exciting. It's something we dreamed of doing for decades, and now it's possible. The flip side of the prediction problem is the design problem. Let's say you wanted to make a structure and you needed to know what kind of sequence could make that structure. And what I'm showing here is a program called RF diffusion or Rosetta fold diffusion, where it works using a denoising algorithm. So if you've seen things like Stable Diffusion and Dolly, these servers that can generate images, the way those work is they take lots of images and they add noise to them and they learn to denoise them. And then they can start with pure noise and create an image out of nothing. Now, what you're seeing there on the left, you can see protein residues that start out as an ideal gas. They're just pure noise. And then we denoise that ideal gas until it comes back to a protein. And we've trained this by taking many protein structures and adding noise to them and learning to unnoise them. And on the right, we're showing one where there's an existing protein and we're trying to create a structure that's going to bind to that protein. This one actually came out of the Institute for Protein Design at the University of Washington, where David Baker is. Chroma is another denoising diffusion model. These are structures made by Chroma. It specializes in large proteins and symmetric proteins and all kinds of things. This is actually coming from the private sector in a startup company called Generate Medicine, the CEO of which they used to be a faculty member at Yale and part of the Rosetta Commons. Their algorithm is a little different in that when they noise, instead of going to an ideal gas, they take the protein, they take the string of the protein, a piece of spaghetti, and they rotate the torsion angles. So you're getting the piece of spaghetti to flop all around. So it takes a protein structure and noises it into a random ball of yarn, and then denoise, and then it can take a random ball of yarn and denoise it into these structures. And these are just a couple examples. All the different labs are working on different types of algorithms. These are two of the fundamental pieces. So going forward from biology, you might take the gene sequence and predict the structure. And from the structure, you might want to predict the function of what it does. If you're an engineer, or if you want to create something new, you might go the opposite direction, design. Start with a function and figure out what structure you would need to perform that function. Maybe you need certain amino acids at the right place to do a catalysis, to do a reaction, to reduce carbon dioxide if you're trying to get carbon dioxide out of the atmosphere. And then you'd have that structure and you try to figure out what sequence would make that structure. Here's 3 examples of different designs that different Rosetta groups have made. The top one is proteins that are made to hold a fluorophore, and so they can fluoresce and make different colors. So that's super useful. You could make glowing plants, but you could also tag things and study things in the cell, study where proteins go. On the bottom left, there's a vaccine for the respiratory syncytial virus, or RSV, made by taking a designed protein cage and then adding lots of antigen on the outside. And then on the right is an example from Sardar Carre's group and at Rutgers of making a material out of protein. He figured out how to assemble these proteins to grow into these fractal tree patterns so you could make interesting materials. If you could imagine that you could control the design of a protein, if you can control what kind of molecule you want to make out of protein, the sky's the limit of what would you like to make out of this. So these come from different labs. The vaccine design, I think, comes from, is that Bill Sheaf's lab? I think at Scripps. It might be Neil King's lab at the University of Washington. There's a few different labs that are doing vaccine design. Okay, so that's a quick tour of the science. We could answer questions about that, but you'll get to pick different labs, have different aspects of the science that they're doing, and you'd be part of this community moving all of this science forward. So I'm going to hand it over to Matt to talk a little bit about what the training program consists of.
Yeah, so the RAMP program is designed to be in person, where each member of the program is in one of these different labs. So we've structured a year program that combines research and professional development. So we're going to walk you through some of the programming that we've developed for the program. It starts with a code school, which will be in person. Everybody will come to Johns Hopkins. And this is a week-long training program where you'll get hands-on training of how to use molecular modeling programs like Primal, and then also foundational skills in Python programming and how to use some of these different modeling tools. And it's set up as a way of introducing these skills in the context of protein modeling and design. From there, the idea is that you would go back to your research labs and get into a research project. So in the research lab, there's the PI, which is the mentor of the lab. And then you'll have a co-mentor in the group, like a postdoc or a senior graduate student that you'll be working with on a day-to-day basis. And the idea is that over the course of the year, you'll work together to come up with a research question and then either do modeling and work with experiments or experimental data within the context of the lab to do real research. So the primary goal is to be a member of the lab doing the research, getting the experience and getting exposure to some of these techniques and ideas and opportunities for applying them. So it'll involve the full gamut of different research parts, from collecting data, analyzing it, writing it, and presenting it. These are some of the members of the community that will be hosting labs. And the projects that they span range from what we work on, which is drug discovery, so using generative models for drug selectivity, to others that are using different types of experimental approaches to inform how modeling works. And then others are trying to use protein design in order to do new types of chemistries. So really a wide range of different problems and questions that they're going after. But the common theme is using protein modeling and protein design in order to guide experiments and discovery of new proteins, new therapeutics, and new technologies. During the year, in addition to the research, we're also going to have opportunities to come back together as a cohort. And so in this case, it will all be virtual because you'll all be in your different labs in different parts of the country. We have monthly virtual meetings where we'll go through journal clubs, and then we'll have different faculty come in and lead different aspects of how to do read a paper, different aspects of what happens if you wanted to join graduate school and how the process goes. The idea is that you get kind of exposure to what would happen if you went further in your career in computational biophysics and where that might go and different opportunities that might be available. But the idea is that come together in these cohort on Zoom, but we also have a Slack channel. And we, in addition to the code school, we'll have a couple of opportunities to meet up throughout the year. We'll come together at least one conference. Typically it's in the winter, along with the rest of the Rosetta Commons community to get connected to others in that community. Go ahead, please. So one of the two big milestones that we'll have through the year is writing a fellowship application for the NSF GRFP. This will give you an opportunity to clarify what the research question is and the research that you want to be doing. And the idea is that if you submit this application and it gets funded, then that opens up the opportunities for pursuing research in a graduate. Typically, if you're admitted into a graduate program, it's funded through the PI, but having your own funding is a great opportunity to have a little bit more freedom and also supports the research in the lab. Once you finish the GRFP, that's in the fall, then we go into geared towards writing graduate school applications. So we'll go through a series of workshops that Stephen will be involved with. And then additionally, there'll be a sort of a peer review of the different statements. We'll do mock interviews and help people pick opportunities in network to find the labs and the opportunities that they want. So the idea is that this program will support moving on to the next stage of the research career. And that typically involves graduate school. Occasionally, others will find research opportunities in industry and where you can help people network into those paths as well. But the program is primarily geared towards graduate school. So in addition to the journal clubs and these different GRFP and the graduate school application, within the lab, there's usually a range of different programming and types of things that are going on, including different lab meetings. You'll be in a department, so there's different seminars, interacting with the other students and working with both your co-mentor and mentor in the project. Part of the program that we're really wanting to emphasize is that we're giving the co-mentors and the mentors a structured program of how to facilitate this mentorship. So like trying to clarify what is the individual development plan, and then training the PIs and the co-mentors in how to do a sort of effective mentoring. And so the idea is that this is a cohort of both the trainees as well as the mentors, and it becomes a sort of a cohesive program. So Jeff showed our meeting that we had a couple of years ago. This is a picture from our meeting last year in the sort of the summer meeting. And then in addition to participating in the different Commons activities, because you'll be there full time, there will be a stipend salary of $32,500 per year. And this comes along with health benefits and this travel funding for going to the code school and also to the different conferences. This should allow you to cover housing and living expenses, and then the opportunities to go on and do further research from that. So I mentioned that going to the winter conference, we have the summer meeting, which I just showed the picture from. This is the winter meeting, and then we just recently had one in Europe. But the winter meeting is timed where you'll get hopefully a chance to present a poster of your project and get feedback from the community about the question, and also make connections among your cohort. I think it's a great opportunity to connect and learn about some of the ongoing research that's going on. And this typically is in March and in Boston. And so there's opportunities to connect with some of the biotech in Boston, which is really the sort of the hub in the US and in the world. The eligibility, because it's an NSF-funded program, a requirement is that people who are US citizens, US nationals, or permanent residents. And then additionally, we're geared towards the program towards providing opportunities for people who may not have a clear path into graduate research. And so in particular, we're looking to recruit people who are either first-generation college or from other under-resourced institutions or have otherwise had less opportunities. Once you put in the application, what we're looking for is the goal to opportunity to go on for the research. So thinking about what type of background do you have, what type of skills are you looking to develop, and how will that prepare you for the next steps? So typically we're looking for people who have either computer science or biology, but maybe want to balance it out. So like coming in with biology, you might want to be focused on developing the data science type of skills. And if you come in with computer science, thinking about trying to ask biological questions. And then along the way, learning computer science skills around how to do data manipulation, how to work with submitting jobs onto supercomputers, how to program in Python, deal with data in different formats, and think about software development and questions around software engineering. These are skills that go beyond just protein engineering and that are useful broadly for bioinformatics, for data science, for other types of machine learning applications as well. So we've incorporated a lot of the new machine learning and generative AI approaches into many of these training opportunities. And so part of that will be understanding sort of the strengths and limitations of how to do generative AI, not just for language models or chatbots, but how to actually use it for scientific applications. If you're thinking about putting in an application, it's relatively straightforward. We've standardized around the sort of NSF format for the ramp. So this includes putting in a resume, having an unofficial transcript from your institution, and then we're looking for personal statements. And this is particularly trying to develop this idea of where are you coming from and where are you going and why is this program going to be effective for you? So why are you interested in it? Which particular opportunities appeal to you? And hopefully we can get that to match up with getting you placed in the right lab. And then how will your research goals be fulfilled by getting these different training opportunities? Additionally, we're going to ask for letters of recommendation. So hopefully this is from people who know you well or ideally can show your potential for research. So if you've done lab classes or had other research experiences, then those would be good choices. And then we'll ask to clarify which labs are the ones that you would think be most appropriate, and that will help us mix and match to different ones. So once we get the applications, they close in March, we'll collect them all, and then they'll go to a review process where we have the faculty on the program as well as representative of past members in the RAMP program. We'll read the applications and evaluate them, and then come to a consensus score. From the top set, we'll invite people to do phone interviews to get a better sense of things that maybe we needed more clarity in your application, and also to help match you up to different programs. And once we have those sorted, then we'll make offers. Say the offer is to come to this lab and do the research starting in the summer for that full year. And then if you accept, then we would go ahead with setting up the arrangements and getting you to become an employee at the institution, and then getting the stipend offset, and then making sure that the travel arrangements are organized for you. Okay, so just the last piece is just in case you're trying to figure out what to put together, there are guidance on creating competitive applications through NSF. So a lot of this guidance applies here. We're particularly interested in people who are really excited about the opportunity and have a sort of clear idea of why that they want to develop these skills and where they can go with it. In addition, trying to clarify what skills you're bringing and which ones that you may want to develop. And then also trying to understand why this program is important to not just biomedical science in general, but specifically for you and why maybe your community or why this is particularly relevant for your personal development and your career path. So I know we're getting a couple questions in the Q&A, but please, we can stick around, and we left a lot of times for questions, and we're happy to go through different aspects of the program and also different aspects of the science that might be interesting. Okay, so just a few more details just to get it out. This is a summary of the different milestones, including the code school, writing the personal statements, the GRFP application, the grad school applications, the interviews, coming out to Winter RosettaCon, graduate school, and then finalizing the research publication or preparation of the final project. And then there's sort of a final retreat to get everything there. So you'll have lots of different opportunities to meet in person, but also we'll be meeting regularly throughout the year. Okay, just in case this maybe isn't quite the right fit for you, for instance, if you're an undergrad and you're a junior and you're going to go back for your senior year, Rosetta Commons also offers a summer RU program that's funded by NSF. So this is a 10-week program that we host in one place, and it comes out for the boot camp similar to the RAMP program, but then you go to different labs and it's just a summer program. So this is geared towards ones that may not graduating seniors, but either ones that are rising juniors or rising seniors. And so this application is February 1st, and it has a similar structure of research and professional development, but it's just compressed into the summer. Okay, so here are QR codes if that's helpful, and we'll post links in the chat where we can get more information about the RAMP and how to put in the application, as well as the research opportunity for undergraduates. And if you have technical questions, Camille, who's on the call now, has her email there, cmathis@jhu. So we'll open it up to questions, and there's aspects that either Stephen or Ashley wants to add as well about the structure of the program or other opportunities associated with this. Maybe we'll have Ashley do a brief intro because she's recently joined and also a valuable member of the team.
Yeah, you guys, I work for the Commons, working on an educational project, RAMP certainly being one of them. RAMP, of course, has its own boot camp and workshop to kickstart your career in protein design. And so I really help organize those for the group in the Commons and oversee those activities. I work with you in the spring on your publication writing process. And so we have recurring meetings on that front to help you get started in your writing, summarizing all of your results and stuff that you've been working on throughout the year. So it's really cool to see everything come full circle. But I know Matt took you beautifully through the program structure, but it's really set up so that you can hit the ground running, get into a lab, start generating data, get some cool results, present them, and then maybe even publish on them. I could see you like, oh gosh. But certainly with a bioarchive or something akin to that, I was really popular in this last cohort as we talked about that. Yeah, that's my role with the program, but I'm absolutely around, definitely a resource. Don't hesitate to reach out with any questions. I think Camille is probably your person for technical questions, but if you just want to touch base with somebody about the program to figure out, is this kind of the right fit for me? Shoot me an email. I'm more than happy to do that. I think it's not too hard to find me, but I'll put my email in the chat so you have that also.
We're getting some questions in the Q&A, and I think we're addressing them, but maybe some of these were some more discussion. I think probably a question that may be on your mind, is this program appropriate? Is it going to be too hard, or is it going to give me the right set of experiences? And I think part of the opportunity that we have here is that people who may not have the right background to make it to be a competitive application in graduate school, but need complementary skill set, or didn't appreciate that there was even such a thing as computational protein modeling until maybe your junior or senior year, that this can be a sort of a bridge opportunity to fill in those gaps or get a better experience for what science it actually involves, and that you can actually get paid to do this. It doesn't cost money to go to graduate school. These are the questions that you may not have appreciated. And getting the year opportunity should give you that sort of more realistic experience for it.
Do you have to work on that project, or can I just go to that lab and work on something else? And that can definitely happen. We'll do interviews. Once we have the application, we'll connect you with interviews with a couple different PIs so you can talk and see if it's a good match. And a lot of times these topics do drift. In fact, the one from my lab, I think, is AI methods for antibody engineering and docking. That's very broad. There's lots of projects on that in my lab. So I tend to talk to somebody and think about, what are you really interested in? Are you more in coding? Are you more into the math? Or do you want to work on particular proteins or antibodies? So I always have a discussion with new lab members to figure out what exactly we're going to do. And that's probably typical for a lot of the labs. You'll have a discussion to figure out what your project will be. So you could certainly, to get more information about possible projects, you could go to the individual investigators' lab sites and look at what papers they've written recently and what other kind of things they're into.
We have a question. How many positions are we looking to fill in 2025? We are looking for 10 fellows to come and join us in August of 2025. And that'll be the largest cohort we have to date.
This is our third year of the ramp, and we did 2 pilot years before that. So it'll be our 5th year of running a post-bac program. And we grew from, we had maybe 4 or 5 the first year, and we've slowly gone up, and it's been really fun. A lot of the students end up going to graduate school. Some stay in the labs that they do their ramp training in, and others go to other schools. A couple have gone to med school or vet school or straight into the workforce. Is this an exhaustive list? So these are the labs that have committed to open a spot for the ramp in 2025. So it's pretty likely that these are the sites where we would quit people this year. And if you have a particular lab that's not on this list that you really want to work with, you could talk to that lab and let them know you're interested in that. If you're able to secure funding from them and work with them, you could join and participate in some of the activities with us and join the cohort. But the ones we have funding for are the ones that are listed in this lab and in this group. Grant says, how does this program complement an industry exit to biotech? I would ask, like, what kind of things you would want to do in industry and do a little bit of research on for that kind of work in industry. What kind of preparation do you need? So I think industry is very eager to get people who know some things about protein design and who know some things about machine learning and artificial intelligence. So there's a lot of opportunities, and I would not be surprised if you had some research experience in this area in an undergraduate degree that you might get scarfed up. But I don't know. I think typically for research jobs in biotech, typically the PhD is the entry-level degree, because that's going to teach you enough time to really get into the field, to read the literature, to know what's going on, to have led your own projects, written your own papers. You have the full skill set to do your own independent research. So if you want to do research long-term in biotech, then a PhD is a good entry degree. I will tell you, in case you don't know, if you go to graduate school in the sciences, you will be paid for that time because you'll be doing research. So typically PhD programs from biochemistry to engineering will have a stipend. The rates are enough to live off of, usually in the place you're in. Hopkins is offering $50,000 next year, which is easy to live off as a graduate student here. You might not put too much money into retirement, and you might not send too much money home to your family, but you would be comfortable while you're going to grad school at the industry after that. But yeah, our program, a big goal of our program is to help people bridge into graduate school. So the professional development is very tailored to what do you need to do to prepare and get your profile together and get the skills together so that you're ready to succeed in a PhD program. So if your main goal is to go work in industry, this might not be the best match for you right away. But if you're okay with doing industry after a PhD, this would be a great program because that's what we'll focus on.
So there's a question on molecular dynamics, Rosetta, and I think you could extend this out to AI too. So Rosetta has traditionally not been a tool that is used for molecular dynamics. There's other tools that are useful. But I think what people are increasingly seeing is that the next broad frontier in protein modeling is trying to understand the structure to function relationship, not just the sequence to structure relationship. And for that, a lot of understanding the different conformational ensemble proteins is really important. So I think it's increasingly important to bring together different tools in order to achieve the different, to tackle different problems. So I think increasingly people are bringing in different skills and different tools to solve problems. Just to reiterate what Jeff was saying is that no matter what research you end up going into, whether it's into staying in academia as a faculty or into industry, as a career, one of the big things that this program is gearing you towards, and graduate programs in general, is to really think independently, to be able to formulate what is the key problem, how do you turn that into steps to solve it, and how do you go through those steps and summarize what you have observed, or how does that lead to achieving the goal? And that sort of independent thinking is really possible to do only through actually doing it. It's hard to do that in a following a textbook or something like that. And so the program is really geared towards that sort of independent thought process, independent developing of ideas. And so you'll work with the lab to formulate the right question and then figure out how to go about solving it. And that skill set, I think, is really valuable no matter where you end up, and whether it's in industry or in academia. But I think going through the full PhD program is typically gets you that roundedness in both technical as well as communication and thinking skills.
Peyton, you asked some experience in bioinformatics, but you've done public health. And I think there's a lot of I get this question from a lot of different people with different flavors. I've done this, but I haven't done that. And this is exactly what this program is for. Our field is super interdisciplinary. I'm in chemical engineering. David Baker's in biochemistry. I think, Nat, are you in bioinformatics? So there's lots of different people from different fields, and you really need to know a lot of different things to get into it. And that's part of why we have this program. You'll come in with some strengths, and we'll help fill in some of the aspects you don't know. So I would say you should have done some computer programming. And if you haven't, on that application tips, there are some links of places where you can do some self-paced courses to get your computing more up to speed. The more computing you have, the better for getting engaged and moving along. It's good if you have a little bit of knowledge of proteins and what they do, but a main point of the program is to help cross-train you in different disciplines. So if you're interested and passionate about this work, you should go ahead and give it a try and maybe try to show evidence of what aspects you've already learned and what aspects you want to learn as you go. All right. Does the stipend adjust to cost of living? It does a little bit depending on the place. Like I know we offer a higher stipend in San Francisco where $32,000 might be a little more difficult, but that varies school to school. Yeah, and you won't know that until you interview with the PIs and we get the final offers out. These are great questions. My favorite thing about this program is the area of the science is so exciting right now. And so we see a lot of young folks who are very excited about learning this topic. And so we get people that are super excited to learn, and it's an area you can just dive in on it and learn so much about. Computer, you can test things really quickly. The AI methods are super powerful. So we're able to do more accurate predictions and more accurate designs than we've ever been able to do before. And so that's really opened our horizon about what we could possibly make. Self-guided computer resources. My favorite is on Coursera. There's a course called Python for Everybody. And it starts with basic Python coding and all different ways of doing Python and getting that general scheme. So that's my go-to when people don't have too much coding to take that course and learn as much as you can. If you know that already, what else could you know? For the machine learning, for the AI, linear algebra, intro to machine learning course, statistics, maybe an intro to deep learning courses. There are intro to AI courses online, also on Coursera, where you can be training a model to recognize different digits. And you can do that in the first hour on that online course. So that gives you some hands-on. Some of these powerful tools are really easy to plug into now. From Python, you just import the toolkits and mix them together. So you'd learn, that would be one thing you'd learn, all about machine learning and deep learning and those things. And you'd also learn the context of molecules, proteins, nucleic acids, fats and sugars, and the biology that goes around it. Medicine, therapeutics. Yeah.
For me, it's been really fun to see just the real growth and development of the people over the time. I started as a new faculty in Michigan, and one of the trainees in our pilot program during the pandemic, I never met him in person because of the pandemic. And then he was able to apply and join our group. And now he's a third-year grad student in our group. And it's been really an amazing trajectory to see how he's come from a chemical biology background and get into doing drug discovery and drug design.
I would say one of the greatest things about the program that I've been able to see is that how students are able to discover what they're made of as a scientist, similar to what Matt just said, but then also their eyes light up in terms of what they can do with all these wonderful skills they're learning. We had talked about going to industry. Some have thought about maybe going to be a researcher in academic spaces. But you just don't know what you don't know before you get into it, right? In this program, what it does really well is that you learn from people from many different walks of life in terms of what's possible. So if you're thinking that you just need just a little bit more information, just a little bit more time to develop your skills, this is definitely a great program to do that. So we really invite you to apply soon.
My favorite thing about this program is that it offers students who might not otherwise have the opportunity to get research experience, to have a year of it, and to also apply to graduate school, because not everybody has that guidance available to write a personal statement or how to do an interview for grad school. So this really offers a lot of preparation for that.
So there's a question from Michael around, what is it more about software engineering or more about the particular modeling? What is it? What is the mentoring structure and how does this work? I guess I can say a little bit about the types of projects that we have in our group. Many of the Rosetta labs have an experimental and modeling component. We're just doing modeling. The types of projects that we would have would be to either do molecular simulations or identify experimental data through collaborators, and then figure out how to bring these data together and use them for, in combination with simulations, to do predictions in different ways. So we're often quite interested in sort of the limitations of modeling going to prospective testing. So making predictions beyond the training data and how to test that. So thinking about doing causal inference, modeling chemical representations, and thinking about the uncertainty and modeling of the data, bioactivity data. And so in this case, the tasks are around a lot of data science. So we often use R and thinking about bringing in data from different sources, and then how to build statistical models and how these can ultimately interact with simulations and different machine learning types of. In other labs, it might be closer to biophysics or biochemistry or chemical biology, and the types of questions they might ask are more about the sort of biophysical characteristics of protein motion and other things like this. So I think different labs have different types of questions and different things they go after, but a common theme is around picking a project that has a mix of developing methods and then doing science with those computational approaches. So what is the question? What are the controls? How do you run the simulations? How do you get the results? And then how do you interpret them? And that scientific thought process is very similar to what you would do in a wet lab situation, except you're exploring the behavior of, so you have to think ways about how to analyze them that make sense. And that is similar to the way you'd run an assay or an experiment in the wet lab, but we're using computers as our model systems for that.
We're happy to stay around a little bit more. If anybody has more questions, or you're welcome to reach out. Probably Camille is your best first contact. She'll be the one fielding applications and answering questions. Thank you all for coming. I really hope to see your applications. I think the applications are already open through that website. You go to the website, has all the information. Then you can go to the NSF ETAP site to do the application. And and I hope to see your applications. I hope to see you in the labs. I hope to see you here at Johns Hopkins for code school this summer. That's when it all begins and when the fun really starts happening. All right. Thank you.
Take care, everybody.
Thanks for coming.
Encouraging.